Hello, and welcome to Taconic's webinar entitled The Laboratory Rat, Biology, Reproduction, and Unique Applications, presented by Dr. Marina Hansen. Please allow me to introduce myself. My name is Megan McBride, and I will be your moderator today. I work in Taconic's product management group, where I am responsible for commercial animal models. Before we get started, I'd like to address a few practical matters. This webinar will be made available to all attendees. We will email a link to the presentation to you by the end of this week. If time allows, directly after the presentation, our presenter will answer your questions as they appear in the chat box. So, if you have a question, please type it in the chat box. The other attendees will not be able to see your question as it is sent directly to the presenter and to me. If you cannot see the control panel and chat box, just single-click on the arrow on the top of your screen. In case your question is not answered at the end of today's session, we will respond to you within the next few days via email. If you are experiencing technical difficulties, please send the host a chat message and our staff will be able to assist you. Let me now introduce our speaker. Dr. Hansen graduated from Iowa State University College of Veterinary Medicine in 2011 and then continued her studies at the University of Missouri Comparative Medicine Program, completing a residency in laboratory animal medicine, as well as a PhD in veterinary pathobiology. She generated a novel cell ablation rat model, which is available through the RRRC. She joined Taconic in 2017 and has veterinary responsibilities for Taconic rodents at several U.S. sites. She also serves as an IACUC member. So, let's get started. We hope you will enjoy today's webinar about the laboratory rat. Dr. Hansen, please begin when you're ready.
Thank you, Megan, for the introduction. So today I'll be talking about the lab rat, mostly focusing on breeding and reproduction. There's certainly a lot to know about the rat, so I tried to keep it focused so that I didn't talk to you all day about my favorite species. So the rat has been a model in research for many years now, pretty much as long as the mouse has. What the rat is mainly used for is a lot of physiology research, certainly pharmacology and toxicology, studies of neurobiology, and I'll speak more to that, and cardiovascular research as well as a small animal model, with pigs being the most common large animal model that's used. So what are the advantages when we're thinking about whether to use a mouse for an experiment or a rat? Most of the advantages of the rat come down to the fact that they're much larger than a mouse. They're both small, but really a rat is about 10 times larger than a mouse. So what this allows is for researchers to take more samples, so serial sampling. They have more blood, so you could collect those samples over a longer period of time. And this might actually let you reduce the number of animals that you use in your experiment, which is in alignment with the 3 R's. Surgical models are often easier in larger species, so vessels are going to be bigger. So if you're inserting catheters or other types of highly technical procedures, the rat lends itself to those models a bit better. On average, they are less aggressive than mouse models. So caretakers do appreciate working with rats once they're comfortable with them, especially when it comes to behavioral research and training rats. Certainly they do operant behaviors better than mice do, which is like lever pressing experiments for drug or cocaine administration. And there are some differences in their serotonin or 5-HT receptors. So I did a little bit of neuro studies in my PhD. So this is an example where a rat could really excel. This is a stereotactic apparatus, and this is to do very precise brain injections. So you can see we have a needle with the rat placed into the ear bars and in a very specific position. We then use the skull suture lines, as you can see in the picture on the right, to find our landmark. And then we use coordinates, stereotactic coordinates off of those landmarks to determine the exact brain position that we're going to do an injection. And in this case, I was inducing a rat model of Parkinson's. So stock versus strain, I don't have enough time to really go through all of the genetics that have happened with rats over the years. Certainly there's not as many strains in the rat as there are in the mouse due to kind of a delay in genetic modifications that were easy in the mouse but not so easy in the rat. So stocks and outbreds have really dominated more in the rat than in the mouse. So stock, remember, we're talking about an outbred model. So that would include Sprague-Dawley, Wistar, and the NIH nudes. Stocks in general tend to have a longer lifespan. We have what we kind of term hybrid vigor. So this confers a higher disease resistance if you are dealing with opportunists or things like that within your facility. One thing I did highlight was usually they have much better fertility as a result. Rats really do not like being inbred. So we can see issues with fertility just as a fact of inbreeding the species over time. Stocks are also usually larger in size than our inbred strains. And then heterogeneity, I start that because even though we are keeping them outbred, they're more heterogeneous than, say, an inbred strain, but they're still not going to be as genetically diverse as, say, the human population is. Strains, we have the Fisher or F344, the SHR, which is the spontaneous hypertensive, and GK rat. And these have been inbred with brother-sister matings for at least 20 generations. And that's when we start to say that we now have a strain. Isogeneity is what rules here because, again, we're fixing all of those genes with each of these generations. So rats come in different colors, albino being pretty much the predominant one. I don't want to go through all of these, but I just wanted you all to have the information of what some of the common stocks and strains are that are in use for rats. And then just highlight that, for example, the GK rat gets type 2 diabetes. So diabetes can have a negative effect on breeding and reproduction. So it's always important to understand that if you're working with a strain, how their particular modification could impact the breeding of that model. Our Sprague-Dawley, however, for example, down here further in the slide, again, very commonly used for embryology and toxicology studies. They're also going to be your most common sentinel for your rat colonies. And then they do have very good maternal characteristics, so they're often going to be the strain you want to use if you need to cross-foster rats from, say, a more delicate strain to a good mom. So you would likely pick a Sprague-Dawley for that. And therefore, also because of that, they're great to use for embryo transfer. So whether you're generating a new model and you're going to use embryo transfer, or if you're trying to clean up a line that maybe has a disease and you would use embryo transfer, Sprague-Dawley would be your recipient of choice. Pigmented strains, so rats also come in great colors like agouti with the ACI. Hoodeds, there are many different hooded lines, so they can have a black hood or a tan hood. But I wanted to point out just a couple. So the Zucker fatty is a black hooded rat that's known for obesity. And then the LEC rat, which is a model for Wilson's disease. And Wilson's disease is a copper storage disease. It's also seen in people. And again, how these phenotypes can impact the breeding is that they either reduce the life expectancy. So what you want to think about is for an LEC rat, they may start to present with the phenotype as early as 5 months of age. So if you're trying to maintain this breeding colony, you would want to make sure that you have enough replacement breeders to allow for the line to continue. So you want to make sure that you always have younger animals within your colony. There's also some lines here too that are predisposed to cancer. Rats are used a lot for neoplasia studies, and so if there's types of cancers that could also impact the reproduction and tumor burden and reduce the lifespan of the rat, then we want to take that into consideration. I just want to give an example of a rat that I love that we have here at Taconic. If you look at the nomenclature, similar to the mouse, when we see the colon in there and you're going to use the lab code first, this denotes that we are working with an outbred stock. So this is the NIH nude rat. It has the same type of mutation that the mouse does. So the FOXN1 in the rat is the ortholog to FOXN1 in the mouse. And this gene is responsible for development of the skin, hair, nails, and immune system. So it has a critical role in formation of the thymus. So the thymus is that organ that tells the T cells what to do and help them mature. That in rats is located up within the top of the chest. And so if they don't have a thymus, then those T cells can't form and they're deficient, so they do not have the normal activity. And then, of course, as a result, because the gene is also important for hair, you get a hairless animal. But they're not always fully haired. Sometimes they can have waves of growth. So just note that if you ever order these animals and they have some sparse hair, that is normal. I bring up this line because it is one that we breed heterozygous by homozygous. And we have to do this because homozygous females are fertile and they can deliver pups normally, but they don't make milk. So then they have trouble raising their litter once they're born. So, and then I should say too, het times home. So we always list the female first when we're talking about breeding formats. So we want to use heterozygous females with homozygous males. And then just to note too, some of you may have had rats as pets. I did when I was younger, and they are different than a hairless rat, and there's hairless mice as well. It's a different mutation, and therefore we refer to these animals as euthymic, meaning they have a normal thymus. So they're not immunocompromised like this model is. Okay, so now I wanted to go into normal reproductive biology, because if we really understand the normal condition in the animal, then it's easy for us that when we have a problem to go back to where we're deviating from normal and try and solution from there. So what I'll cover is everything from sexing the animals, being able to tell male from female, through puberty and pregnancy and weaning, and then also, as I touched on a little earlier, reproductive senescence and knowing when you need to turn over breeders within your colony. And again, we need to know these because if any of these deviate, then that's when we can start to see reproductive failure within our colony. So sexing, like most rodents, we're going to be using anogenital distance. So the distance, as you can see here with the black bars, is going to be much shorter in a female than it is in the male, because you're going to have room for the scrotum there in between. Males, many rodents have what we call open inguinal canals. So that inguinal canal is where the testes actually descend from the abdomen into the scrotum. And that stays open. So when you pick up a mouse or a rat by the tail, you can actually see them retract the testes. So just because you don't see testicles doesn't mean that it's a female. So we really want to use anogenital distance. And the other key indicator in mice and rats is nipples on the female. So if you pick up an animal and you see nipples, and you know that's a female. So male reproduction, as I said, they have those open inguinal rings. And here is a male. They are This one's pretty obviously a male. They have very large testes for their body weight. You can also see in this diagram here of the urogenital system, they have a lot of accessory sex glands that help with the formation of the vaginal plug, for example. And so you just want to be aware that they have some of these other structures in here and that they're normal. It can be a little confusing at first if you're doing dissections to tell what some of these organs are. Guinea pigs are the ones that tend to trip up a lot of people, where the coagulating, the seminal vesicles of the guinea pig can look a lot like uterine horns. But so if you really understand the normal anatomy, then it's much easier to see when something might be abnormal in the animal. As far as development of the male, when they're going to reach puberty and start producing sperm, is about 45 days, but really they're going to get max production when they're about 75 days old. So if we're talking about collecting males or setting them up for an artificial reproduction procedure like sperm collection or rederivation, we would want to be in that max range. And this is really numbers given for, a lot of them are based off of the Sprague Dawley or the Fisher. So there is going to be some strain variability. Females mature a little bit earlier here, so they'll have the vaginal opening occur at 33 to 42 days, or about when they're 100 grams. So for strains that are more inbred, where they might be smaller than your outbred, this 100 grams might occur a bit later. And so after the vaginal opening occurs, 1 week later, they're going to have their first proestrus. So proestrus is at that start where they're going to be receptive to the male and then begin their estrus cycle. Estrus cycle is pretty similar to the mouse. It's 4 to 5 days long. And they do have what we call a fertile postpartum estrus. And what this means is that if you keep the male in with the female, after she gives birth, she will ovulate and come into heat or into estrus right after she gives birth. So this lets you be able to breed her back right away and have another litter while she's still nursing that previous one. So right when you would wean her first litter, she would have that second one born. They do have 2 cervices, so this differs from the mouse. So the mouse has a single cervix, and I've listed the NSET here. So that's the non-surgical embryo transfer. So in mice, there's been some work done to, instead of doing embryo transfer surgery, you can just implant the embryos with this device transvaginally. This is not as effective in the rat because, again, 2 cervices going up to each of the 2 uterine horns. It's a little bit more technical than the mouse. So not as successful as it has been for the mouse. Lastly, and this is important especially because of the types of neoplasia rats get, but they do have 6 pairs of mammary glands. And what's really interesting to note is how extensive this mammary tissue is. So you can see it goes all the way up to their neck and even around and up onto their back a little bit. So you might see a tumor that's up on the neck or the shoulder even, and you might think it's weird that that could be a mammary tumor, but remember they have this extensive mammary tissue. So it is still possible to have mammary tumors that seem like they're on the side or on the back. So estrus cycle and mating, as I said, it's 4 to 5 days long, and this comes right out of the rat book reference. And vaginal cytology can be a really useful tool. It does take some training and experience to get used to reading the slides and interpreting, because even though these pictures look pretty representative, there's a spectrum, right? So as they move from one phase of estrus to another, there could certainly be some slides that might take you a little bit longer to read and interpret. But so here we have the 4 phases. So there's proestrus, estrus, medestrus, and then diestrus. And you can read here what the behavior is. So estrus is the one where we're going to have the female be accepting of the male and when we're going to have ovulation occurring. So you can take a vaginal smear, put it onto a slide, and then look at these cells that are present at each of the stages, and this would help determine when she would be receptive to a male. I did also note that the mouse pheromone effects that we see, so if you're familiar with the Bruce effect, the Witten effect, that are these different pheromone and signals in the animals, are not really known to be observed in the rat. But pheromones are important, at least for them just to be able to smell each other, to just have that courting behavior in the rat. So we do find that in most strains, copulation occurs during the dark phase, with ovulation occurring between usually midnight and 2 a.m., depending on how you have your light cycle set up. So the next morning, they're at about 0.5 days post-fertilization. And when people are doing embryology brain mapping, that's usually the timing that they're using to determine what map to read off of. The plugging is a bit different in rats versus mice. So the plug tends to fall out of the female rat much faster than it would in the mouse, and it can be much harder to find. So in general, we say that just because they don't have a plug doesn't mean that they weren't bred. If you combine plug checking with vaginal smears, that can increase your chance of confirming pregnancy. And visualizing sperm is certainly a good indicator that she was bred and could likely be pregnant. Sprague dollies will have conception rates of greater than 85%. So this is a pet rat, that's why there's no gloves in the picture, but I thought it was the best picture of a nice gravid or pregnant female. So you can see her nice round belly. She's probably due any day to have some pups. So as I said, determining pregnancy, we can certainly do a swab and look for sperm within the female, and that's a great sign that she could be pregnant. Palpating, some people are really good at palpating, and they could even palpate before 12 days, but usually by 12 days you can start to feel that there's some fetuses within the abdomen. Gestation is anywhere from 21 to 23 days. In some cases, it can be a bit earlier at 19 days. It can be a bit strain dependent, and then also depends on when the embryo will implant. They don't implant into the uterine lining for a few days, and that could be extended just depending on other stressors in the rat. We can see a delay, as I said, if she's also nursing another litter, then she'll delay implantation, and so you'll see a longer interbirth interval. They don't really build nests like mice do, these nice big dome structures, but you can give them other types of nesting material. They still like to have enrichment, just don't expect them to build a nest like a mouse. And for all the things we know about animals and biology, one thing we still haven't figured out in, I think, any species so far is how parturition or birth starts. So we don't really know what the stimulus is, but once she gets going, usually labor takes 1 to 4 hours. And we tend not to see dystocia or difficulty giving birth in rats, so fairly uncommon. So you might see some blood back there, or as she delivers some of the pups, you might see it, her deliver one and then take a break and then deliver the rest. That's all variation of normal. Now we have the pups on the ground, so lactation will kick off without the suckling reflex, so she'll have milk when the pups are born. But in order for her to continue lactating, she's going to need the pups there to continue suckling to give her that stimulation and get the prolactin. Peak lactation occurs about 17 days postpartum, which makes sense because if you've ever seen rat pups, they grow really fast, and that's close to weaning. They suckle very frequently, which adds up to about 18 hours a day. So you can imagine mom gets pretty tired of her pups by the end. And they are born, this is what we call altricial young, so they're born with their eyes closed and hairless and pretty helpless. So this is in contrast to guinea pigs, which deliver their young precocious. So they come out with hair and eyes open. As we increase litter size, birth weight of the individual pups will decrease. There's only so much room in the uterus, so that's what happens as we have more pups. And then you can see here some of the timeline for when key developmental factors happen in the pups. So they'll pretty much have their hair by 7 to 10 days of age, and then we're going to wean them at 21 to 23 days. Some of our inbred strains might need a few more days with mom. Senescence, so here's a picture of a fat rat. So certainly when they get obese, the males especially are not going to be as interested in breeding. So you might see fertility drop in those animals, especially on an ad libitum diet. Females, usually by 7 to 9 months, we can see a drop. And again, this is usually going to be for outbreds or our more common rat strains. It could be very different for an inbred strain. And males here, I have some numbers listed. So Fisher, we say about 18 months for a male, and Sprague Dolly for 2 years. I don't recommend keeping a Sprague Dolly male for 2 years because again, at that point, we're talking about obesity and other factors, cancer, that could reduce their lifespan. So factors that influence breeding, I'll just cover these pretty briefly, but I want you to kind of look at it as an effect of the environment that can impact breeding and then largely genetics. And what can happen here is if anything is just a little bit off in your environment, a Sprague Dolly might perform just fine under those conditions, but once you start breeding a genetically modified line is where you can really get into this warning overlap area where it presents now as a bigger problem. So I'll go into some of these things, and I put diet and handling under both because they're kind of intertwined. Some animals like the LEC rat, for example, needs to be on a low copper diet. So that's kind of a combination of altering their normal diet that they need with the genetics. So lighting is a big one. Certainly there's been papers that have reported both continuous light and then interrupting the light having an effect on the ovaries. And if you have ovarian atrophy, which is basically loss of that tissue, then you can imagine they would have trouble having pups after that and getting pregnant. Noise and vibration, there's been, over the last years, a big push for using IBC racks, but remember they have motors on them that generate a lot of noise and vibration. We speculate that it's most times the vibration that disturbs the animals more than the noise due to the hearing range that rats have. But you do want to consider, the noise and vibration that any equipment, radios or alarms in your facility could have on the breeding. And I just have a picture of a bat detector because this can pick up high frequency sounds that would be out of the range that you could normally hear, but that rodents can hear. And then litter size is another factor that can influence reproduction in that animals that were born from larger litters with lower body weights also had delayed testicular maturation, so they didn't come into puberty as early. So you could see a delay in some litters if you're setting up males that fall under that category. Diet, we have well-formulated rodent diets. I think rats have probably been the species that we have the most nutrition data on. So their diets are pretty whole. But if you're working with a diet that maybe is nutrient deficient because that's what the research requires, or if you have a process where maybe your autoclave is malfunctioning and some of those nutrients are out of spec, then you could see undernourishment during pregnancy. And as in most species, if they're undernourished during pregnancy and lactation, then that would affect that litter. But what's interesting is that it also affects the reproductive performance of those pups when they get mature. Additionally, you can just prolong that lactational diestrus, and that's the period where while they're lactating, they're not cycling. So you can actually make it longer, and then it would take them longer to breed back and get pregnant again. There's been interesting studies done on caging and housing density because of the change that was made in a guide to give more space to females with a litter. So our recommendation and what the literature has shown is that monogamous continuous housing seems to provide the best reproductive results. Trios, there's really not enough space in most of our rat cages to keep trio mating. And even pulling the male, as you can see here, there's just one female in a cage with pups was the best outcome for the litter survivability and welfare. And what this paper was using here is this press posture behavior. So this is where the female is going up to the side of the cage and pressing against to kind of get away from the pups. And this was considered a stress response. So if you see this, the indication here is that she's stressed. Usually you see it more as the litter ages or if there's more animals within the cage. Remember that anytime you are going to be altering the cage density, you want to look at, again, your cage change frequency to make sure that we're monitoring the moisture within the cage and the wetness, as pups obviously can be susceptible to hypothermia. Similarly, temperature, they've done studies that show common in other species as well, high temperatures are not good for sperm. So we've seen male infertility even as low as 26.6 degrees Celsius. So moving on to breeding management and solutions. So we've gone over normal and things that could influence light. So hopefully now you're thinking about, okay, I have an issue, how can I kind of work through this? So what I wanted to give you was just, okay, I have a case of reproductive failure. How can I use normal to now recognize where we might be off track? Surprisingly, sexing is a great place to start. So please make sure that you have a male and female together in that cage. It might seem trivial to do, but it has happened before where researchers are wondering why they don't have pups and somebody had placed two females together. Puberty, so we want to, it's good to keep a record of the time to first litter, especially if you're working with any new inbred strains, to keep track of how long it's taking them to drop their first litter or look pregnant. The estrus cycle, we talked about vaginal cytology, so you could check how long they should be in each stage and see, like, okay, does it seem like they're spending more time in diestrus than they should be? Are they having a normal cycle? Is everybody coming into estrus at least every 4 to 5 days? Mating, so if you are doing time mating, certainly this becomes more critical, but documenting that mating did occur either through sperm or plug checking. Just remember again that the plug is not as reliable in the rat as it is the mouse. Pregnancy, it's a great idea to just note who looked pregnant, because then if you never get pups out of that female, then that would lead us to think that maybe we have an embryonic loss happening, that she can't support a pregnancy. And the interbirth interval, so that's the time from one litter to the next. So is that prolonged? Are we taking advantage of that postpartum estrus and tracking that as well? Gestation, is it within that normal range that we would expect for a rat? How many pups? And then again, going back to that embryonic loss, did did we get the number of pups we expected? And then if they're genetically modified, did we get the genetic ratio we expected? So did we not see any home animals out of this, homozygous animals? Cannibalism, very rare in rats, but it can occur. Usually what I've seen first is that the pups fail to thrive and then mom eats the dead pups, not necessarily that she actively kills her live litter. And that can be hard to know. A lot of times it happens when we're not in the room to observe it. So good maternal care. Remember, Sprague Dolly typically are very good moms, but there are some other strains out there too that do a great job as well. So we want to look at, okay, how many, try to minimally disturb the animal, but assess how many pups there are and how healthy they look. Do they have milk spots, which is pretty easy to see in our little hairless neonates. And then assess if we have what we call pre-wean mortality. So before they get weaned, how many pups were lost. And then when it comes time to wean them at that 21 to 23 days, how many of them did she successfully wean? And then one thing when we're managing the whole colony is, are we set up for success to keep this colony going over time? It can be very common to get excited that you have pups and you can do your experiments and you forget to plan that some of those pups need to be your breeder for the next generation. And tracking generations is important as well, especially with transgenics, where we could see a change in the phenotype over time. So it is good to track generations as well, but plan for those replacements and plan them to turn over in a more regular pattern instead of like an all in, all out. So if you kind of always have a small percentage of rats ready to replace the older ones, then that ensures that you'll have a steady supply that you can meet your research needs. So I just wanted to give you a quick example of what breeder books could look like. Certainly you can use computer systems that can do all of this tracking for you. Colony management software is available, or you can use just a standard lab notebook and keep track of this. So one technique is to keep a breeder book and a wean book. And you would have all of your information, like what your breeding scheme is. So you have them set up in pairs, or are you putting the male, moving the male around? Maybe you only have one founder, and so you want to breed him to multiple females. So maybe we call that jumping from one cage to another. Are you breeding to get homozygotes? So is it a het by het mating format, or are we using wild type animals that you order from a vendor? So the breeder book would keep track of all of the litters that you have, and there you would list what breeder cage they came from, the strain and the genotype of your female and male, and then when you set them up. And then your cage card would have information as well that you would document. Maybe you're tracking, who looked pregnant right on the cage, so you could see that quickly. And then in your wean book, the litter number corresponds. So you would always be able to tell that these pups, so pup HT301 on the first line here from the litter, came from litter 2. So I know the mom was EE703. So this lets you keep detailed generation information on your line in a pretty quick way. But as I said, certainly there's computer programs that can do the same. So I hope this has been helpful for you to manage breeding rats, and wishing you much success. So again, it's focusing on normal, keeping the proper records, and then understanding what to troubleshoot and what to work through first when there's difficulties happening within your rat colonies. So I did just want to touch a little bit relatedly to breeding, is that rats get a lot of neoplastic or cancerous diseases. And so these are ones that you can see show up in your colony that again could impact the breeding. And I just wanted to highlight that Fisher rats, for example, can get some tumors. Just about all rats will get the benign fibroadenomas. And then vaginal septae, so that's where you'll actually get a membrane that could prevent the rat from getting pregnant in a Wistar or Sprague-Dawley that's been observed. So just keep those in mind. And then if all else fails and you're out of options with natural breeding, there are some assisted reproductive techniques that we've used in the rat. So I've done, this is my study on electroejaculation in the rat. It's not a great idea for artificial insemination because you get a lot of the coagulating fluid in there as well. But IVF can somewhat be done in the rat, but there are quite a lot of challenges to overcome. So in practicality, what most labs do is embryo cryopreservation. But there are a few labs that have the capability to do sperm cryopreservation, with the outcome being that they can then take the sperm and do a process called ICSI, or intracytoplasmic sperm injection. And this is where they actually take the sperm, the head of the sperm, and inject it into the oocyte to fertilize the egg. Because again, doing IVF with frozen rat sperm is typically not successful as it is in the mouse. So I just suggest that you consult with people that are proficient in these techniques if you do need help. And with that, I will take any questions. And here is the reference on the laboratory rat, where you could get not only more information on reproduction, but rat behavior, pretty much anything. This is a good resource for you to go to.
Thank you, Dr. Hansen, for that fascinating presentation. If you have a question, please type it in the chat box in the control panel. We have already received some questions from the audience. Our first question is about enrichment and what types of enrichment you would recommend for use with rats.
Yeah, that's a great question. So as I said, rats don't nest as much as mice do. So usually nesting squares, sometimes they might shred them a little bit, but they don't really fluff them up like a mouse will. But items like paper towels or crinkle paper, and even using like a shack or a hide are really good options for rats to use.
Okay, you mentioned some dates that were appropriate for weaning of rats. If you don't happen to know when the litter was born, can you talk a little bit about signs that would indicate the rats are ready for weaning?
I think your best bet is to track when the litters are born. So ideally, be checking for new litters each day to keep track. And then that would give you that determination that you're at the 21 to 23 days point. They should be up and moving around the cage, very active, able to drink water and get food from the feed hopper. And then your institution with your veterinarian might also establish a weaning weight, so a minimum weight that the animal would need to be weaned.
We didn't talk very much about diet or water. Are there specific recommendations you would make for diet and water type or water treatment for breeding colonies of rats?
Yeah, so some believe that for diet, having a little bit more fat in the diet promotes breeding. The caution there is that adding more fat to the feed will make the rats a bit fatter. So there is some risk there. But usually a 5 to 6% fat diet works well for breeding. Water, it's really going to depend on the health status of your facility. Your water could be acidified, it could be hyperchlorinated, or autoclaved, especially if they're immunocompromised. At a minimum, I think most facilities do testing for things like coliform counts or minerals within the water.
Can you talk a little bit about the different methods used to determine pregnancy in rats, and in particular, which method Taconic uses?
Sure. So you can use palpation. That takes a little bit longer. You can do ultrasound, like we do in other species. But ultrasound usually isn't effective that much earlier than just palpating. Maybe you can do it a couple days earlier. For real early detection, it's not 100%, but certainly, as I said, taking that swab and looking for sperm can give you a really good indicator that she was at least bred. Vaginal impedance has been used in the past with one of those impedance meters. So you just insert that into the vagina and you'll get a couple readings. We used to use that at Taconic, but we found it to be a bit inconsistent, and the technicians were really good at doing the vaginal cytology. So currently now we do all vaginal cytology and check for presence of sperm and then grade females accordingly.
4 times matings. How does Taconic determine embryonic day?
Yeah, so if we set them up on day 0, then the next morning, that next day, would be the first embryonic day. Your neurobiologist might be a little more specific and say, you know, day 0.5, but we will call it day 1.
We're going to switch gears a little bit and talk about shipping. Is it okay to ship pregnant rats?
So it can be in a lot of cases. You always want to be cautious with a strain that you've never shipped before or one that might have other phenotype issues. So I would say it's better to avoid it if you can, or at least avoid early gestation and late gestation. So early gestation, again, remember I said the embryos might not have implanted yet, so that could be a risk for early embryonic death. And then late gestation, what we worry about is that they could deliver in transit. So we don't ship females later than embryonic day 18 for fear that they would deliver in shipment.
We have time for one final question. You talked a little bit about the comparison between mice and rats, and one big difference is really the extent of use of genetic engineering, right? We know that there's thousands of mouse genetically engineered models. Can you talk a little bit about genetic engineering in rats and what the current state of the landscape is?
Yeah, so where rats got behind is when it came to ES cells. So a lot of early technology in the mouse was ES cells because we had the 129 and we knew how to do the ES cell work quite well. Rat ES cells are quite a bit more finicky, so it took us a long time to figure out how to do homologous recombination in rat ES cells. Certainly transgenics have persisted, and we've worked out all of the issues with pronuclear injection and handling rat sperm and all of that. But what's really happened now is with the advent of CRISPR and these other technologies, we've been able to generate the rat knockouts that we weren't able to before. So my hope is that the rat is going to catch up. Unfortunately, I think some people that were looking for that specific model have switched to the mouse and just learned how to do some of those procedures or techniques that were traditionally rat techniques in the mouse. And with the caging cost being a bit higher for a rat, sometimes it could be hard to get now a mouse researcher to consider using a rat. But certainly, I think there's still utility for the rat, are still heavily used, just not as much as the mouse is today.
Thank you again, Dr. Hansen, for that exciting presentation. In case your question was not answered during today's session, we will respond to you within the next few days via email. In addition, we will email a link to the presentation to you by the end of this week. Thank you all, and have a good day.